Hi everyone, let's continue with the revision of carbohydrate chemistry. First question, compare and contrast starch with glycogen. So you need to explain what is starch and what it is composed of, and also about glycogen, then how it is being affected by various enzymes. Same with starch also you need to explain. You should give equal importance for both, that is 1 mark you need to explain with starch, and for 1 mark you need to explain glycogen. So starch is from carbohydrates from plants, mainly from cereals and vegetables. Then it consists of amylose and amylopectin. Then how it treats with the boiling water. Then what is the linkage? Amylose is made up of glucose units with alpha 1,4-glycosylic linkage. This is the linkage which is present in starch. And next, when beta amylase acts upon amylopectin, maltose units are liberated from the end branches of amylopectin until the action of enzymes is blocked at 1,6-glycosylic linkage. So the insoluble part absorbs water and forms paste like gel, and this is called as amylopectin. And amylopectin is also made up of glucose units, and it is having alpha 1,6-glycosidic linkage. Then when it is hydrolyzed, the starch is hydrolyzed, it forms a blue colored complex with iodine, and it disappears. It disappears on heating and reappears with cooling. Then next is with the action with the amylase. Starch, when it is coming in contact with the salivary amylase, salivary and pancreatic amylase, alpha amylase, which act random on alpha 1,4-glycosidic bond, splits starch into smaller units called as dextrins, and this finally forms, after the splitting up, it finally forms alpha maltose. So that is action upon amylase enzyme. Next you need to explain about the glycogen, that is the reserve carbohydrate in animals. That is, glucose is stored in our body as glycogen. It is stored in liver and muscles. About 5% of the weight of the liver is made up of glycogen. Excess carbohydrates is deposited as glycogen. Glycogen is composed of glucose units joined by alpha 1,4 links in straight chains, and also alpha 1,6-glycosidic linkages. So you need to differentiate, compare with the linkages and how it is acting upon with the help of enzymes. Glycogen is more branched and compact, whereas starch is not branched. Next question, why starch can be utilized by humans but not cellulose? Starch is composed of amylose and amylopectin, and its action on amylase, it is broken down into smaller units, dextrins. Whereas cellulose, it is the supporting tissue of plants, and it is having glucose units combined with beta 1,4 linkages. It has a straight line structure with no branching points, and beta 1,4 bridges are hydrolyzed by enzymes cellulobiase. Cellulobiase is the enzyme which is hydrolyzing the 1,4 linkages, and it is not present in humans or animals. It is absent in humans. Human digestive enzymes do not contain cellulobiase. That is the reason why it is not being digested in our body. Next question, example of dietary sources of cellulose. That is all leafy vegetables. Dietary sources, leafy vegetables. Next question, what are glycosides? Mention one example. So you first need to give the definition for glycosides. Glycosides, they are hemiacyl group of when the hemiacyl group of monosaccharides is condensed with alcohol or phenol group, it is called as glycoside, and it is non-reducing with Benedict's reagent because the sugar group is masked, and they may be hydrolyzed by the boiling with dilute acids. And examples, any one example from the column you can explain. Next question, Why sucrose is a non-reducing sugar? Sucrose contains glucose and fructose. Sucrose is not a reducing sugar, and it will not form ozazone. This is because the linkage involved in first carbon of glucose and second carbon of fructose and free reducing groups are not available. And the linkage present in sucrose is alpha beta 1 to glycosidic linkage. Next question, invert sugar. Again, invert sugar, you need to explain about sucrose. Hydrolysis of sucrose will produce one molecule of glucose and one molecule of fructose. Therefore, the product will change the dextro to levo, or plain rotation is inverted. So, equimolar mixture of glucose and fructose is thus formed, and it is therefore called as invert sugar. Next question, name the reducing substance excreted in urine, that is glucose, fructose, lactose, uric acid, ascorbic acid. Any one you can, any few you can mention. Next question, Benedict's test. Benedict's test you have already done in your practicals, and all those practical knowledge you need to apply here. That is, what does Benedict reagent consist of, what are the compositions in the Benedict reagent, and the procedure, and what are your findings, how the color changes. Benedict's reagents is commonly employed to find out the presence of sugar in urine, usually done in diabetic mellitus. Then the reagent consists of sodium bicarbonate, copper sulfate, sodium citrate. Then how the procedure, 5 ml of Benedict's reagent. Then why the color and what each color indicates, why the color is being formed, all those things you need to explain. Next question, define epimers and give 2 examples. When sugar are different from one another only in configuration with regards to the single carbon atom other than the referent carbon atom, they are called as epimers. For example, glucose and mannose. Epimers of D-glucose is D-mannose and D-galactose. Next question, mutarotation. Mutarotation, when D-glucose is crystallized at room temperature and a fresh solution is prepared, its specific rotation of polarized light is found to be 112, but after certain hours, 12 to 8 hours, it changes to plus 52.5. So, this change in rotation is called as mutarotation. Thank you.